Chapter 14, Section 3, Photosynthesis and Chlorophylls. Now, I know you're sitting there thinking, wait a minute, this is microbiology. Why are we talking photosynthesis? Photosynthesis in plants. No, you're wrong. Photosynthesis isn't just plants. Photosynthesis is any organism that can utilize light. Light to power the biosynthetic or the anabolic processes to synthesize organic compounds. Done. Fini. That's it. Yeah, we know it mostly from your intro cell biology, plant biology as plants, but plants have only been around a few 100 million years. I'd say, maybe what, 600-700 million? Beside the point. Bacteria have been around longer. Archaea have been around longer. They've been doing it longer. So we're going to be talking about organisms that, yes, they are, some of them are going to be photosynthetic, known as autotrophs. Specifically in this section, we're going to be focusing on the autotrophs. Photo uses light, photons of light, to power carbon fixation. Okay, they're going to take CO2 from the environment and they're going to link it 3 by 3 by 3, just like you've seen in previous courses for photosynthesis. 3 carbon sugar, 3 carbon sugar. Link those 3 carbons. You can make yourself a 5 carbon, a 6 carbon, whatever you need. If you don't want that, then you can link them together in certain ways to make amino acids. You can link them together to make nucleotides, to make fatty acids, whatever. Now, not all phototrophs, not every organism that's going to utilize photons of energy is autotrophic. There are photoheterotrophs. Okay, they also use organic carbon. Okay, this is going to be carbon that's in small molecules. Okay, not CO2. So these are going to be things that have to be ingested by the cell, taken in by the cell. The energy to break things down and whatnot for ATP synthesis to power the breakdown, the catabolism, which can then be turned around and utilized in the anabolic methods. Well, that's going to come from light energy. Take in small organic, link them together, break them down, carbon, 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 individual carbons, link them together. Use photonic energy to power that. So as I said, photosynthesis originated in bacteria. Now, how bacteria have been doing it for the past couple of billion years may not be exactly how you learned, how I taught some of you in intro cell and molecular, or how some of you learned it in cell or plant bio. Got to remember, it originated in them, so they're going to have, they've been around a lot longer, a billion years longer, so they're going to have proteins that are analogous, but are not exact copies of what you see in eukaryotic cells. The light reactions, which we'll talk about more in a few slides, remember, this is nothing more than the capturing of the photonic energy. Not the photons themselves, just the energy in that photon. Capture it, utilize it to make ATP. Set up a proton motive force, pump some hydrogens, so those hydrogens can flow back through, ATP synthase, giving you lots of ATP. A lot of ATP that is going to be required for the light-independent reactions. Notice here I'm not saying the Calvin cycle. Notice I'm not saying, not using the term Calvin cycle because it's not exactly Calvin cycle, depending upon the microbes you're looking at. Also not saying the dark reactions. Ooh, you ever want to piss off a plant biology person? Say the light reactions, the dark reactions, their head will explode. Now, the light-independent reactions come in two flavors. Two flavors we're going to focus on: oxygenic and anoxygenic. Oxygenic means they're going to be splitting water. Remember, whenever the pigment gives up its excited electron, it has to replace that electron. Thus, that splits water, two hydrogens / electrons, one oxygen. Well, that oxygen's a byproduct. Breathe deeply, people. There's where it's coming from. Anoxygenic photosynthesis means they're not splitting water. They're getting that electron from somewhere else, some other molecule, an organic molecule, an inorganic molecule, doesn't matter. It's going to be different species to species, but it's not water. Thus, no oxygen will be given off, hence anoxygenic. All photosynthesis is going to have some pigments. Okay, the purpose of these pigments is not just to make things colorful. It's the pigment, what we call a pigment, the molecule we call a pigment, that absorbs the photonic energy of all wavelengths except one, which is what it bounces back, reflects back, which is what we see and we perceive as green, purple, whatever. Chlorophylls, you're going to see these plants, the algae, the cyanobacteria, and then bacterial chlorophylls. These are going to be the ones you see in anoxygenic phototrophs. They look similar to chlorophylls, but a lot of the functional groups will be moved around. Different functional groups, which we'll see in a few slides. So when you compare side by side, anoxygenic, oxygenic. Oxygenic is what you've learned in other courses. Photonic energy is going to be utilized. Capture the photon's energy, excited electron, send it up the reaction center, pass it on, blah blah blah. To replace that excited electron, water is split. The two hydrogens / electrons with proton baggage will be utilized they will replace. The ones that were kicked up because they're in a high energy state are going to be utilized in the fixation of carbon dioxide into some form of sugar. Those excited electrons that are going to be bled off are also going to be utilized. Some of that energy is going to be utilized here in the process taking ADP, making ATP. And then that's going to be kicked over here along with those excited electrons, because remember, NADPH, NADH, FADH2, those excited electrons bringing in a lot of reducing power, so you're going to be able to fix multiple CO2s, come up with sugars. But notice right there, oxygen is given off. Anoxygenic, notice here at no place do you see water being broken and oxygen being released. Electrons are excited in reaction centers. The excited electrons kicked off, taken over to a cytochrome complex to be bled off to pump hydrogen, set up proton motive force, ADP to ADP thanks to ATP synthase. Well, those excited electrons have to be replaced, so you're going to take hydrogen sulfide, turn it into sulfate. In the process, redox reactions to replace the electrons. CO2, atmospheric CO2, will be fixed to make sugars. Again, notice nowhere here does it say O2 is being released as a byproduct. The chlorophyll and the bacterial phylls, okay, they're based on what is referred to as a tetrapyrrole. Okay, pyrrole is a carbon ring. Tetra means it's 4 rings held in place, and at the center of where those 4 rings intersect, you're going to have a magnesium. If we were talking cytochromes and things like that, you would have the tetrapyrrole, but it would have an iron center. Think hemoglobin. Kind of the same thing, structurally wise, not metabolically, not chemical reaction wise. Chlorophyll A, or a version of it, is going to be our principal chlorophyll in oxygenic photosynthesis. Okay, chlorophyll A has been around a long time. Okay, this is what makes plants, algae, some bacteria look green. There's different versions of these chlorophylls, different versions of these bacteriophylls. They're each going to have, because of their structure, going to have a slightly different absorption spectrum. Where they have their peaks, where they have their valleys is going to be slightly different based on shape. When you look at the shape, you can compare chlorophyll A and bacterial chlorophyll A. Here's our rings. I said 4, it's multiple, sorry. 1, 2, 3, 4, 5, you go through. 1, 2, 3, 4. These rings. So it's this 4-sided ring thing. So I got the 4, sorry. All of these groups you see here, these functional groups, will be changed, which is the difference between chlorophyll A, chlorophyll B, between the different bacterial chlorophylls. Here, chlorophyll A, chlorophyll B, yellow and green are going to be our major differences. I mean, you're down here, the phytol and the cyclopentone ring. These are similar, but it's right here. These groupings, or our difference are, which means we're going to have different absorption spectra. Remember, I told you peaks and valleys at different wavelengths. The different types of these bacterial chlorophylls, right now, there are, as far as I know, there are 7 different ones. Here is the base tetrapyrrole ring structure, and our 1, 2, 3, 4, 5, 6, 7. Where these are, here's the difference. Our 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7. 7 different A, B, C, what is that? I can't read the sub. C sub S, D, E, and G. Each of these different R groups for these is going to have a slightly different composition, different shapes, different functions. As you can see here by, the absorption, the pigment, purple, purple, green, green, and its absorption maxima, 740, 745, 830, 1020, so on. Now, just like we've seen in plants, you talked about in other courses in plants, the chlorophylls and the bacterial chlorophylls are not free-floating. Okay, they are anchored in membranes. They're anchored membranes in close proximity together, forming what we commonly refer to as photocomplexes. Okay, this way, all the proteins that have to function together are held in place together. The photocomplexes, you're going to have a reaction center at the middle of them, surrounded by pigments, the chlorophylls or the bacterial chlorophylls. These pigments are acting as antenna. The whole point of antenna is to capture something, signal, light, whatever. Well, that's what these guys do. They're acting as, word here. They're forming this ring around a reaction center. This is what's going to capture the photons of energy, and it's going to go from, light harvesting complex 2 to 1 to the reaction center, which will then kick up an excited electron, which will go to an electron transport chain of some sort, where the energy will be bled off, pump hydrogen, set up a proton motive force, and so on and so forth. Now, In eukaryotes, plants, algae, you're going to have chlorophylls. Remember, think to your endosymbiont theory. The chlorophylls at one point in time, almost 1 billion years ago, were green pigmented archae prokaryotes that became symbionts, that evolved to become endosymbionts. So we have the chloroplasts, and if you think back, your chloroplasts had dual membranes and the thylakoids inside of them. Okay, remember that was the green pancakes. Thylakoids are surrounded by the stroma, which is the aqueous solution where a lot of those Calvin cycle or light independent reactions would occur. Now in prokaryotes, since they don't have internal membrane organelles, we're going to see that this is going to be different. In prokaryotes, in the purple, the green, purple bacteria, the cyanobacteria, the purple bacteria, and some of the archaea that are photosynthetic, there's going to be invaginations of the cytoplasmic membrane. So instead of being a nice round circle, there's going to be parts of it that come and fold in and stuff like that, forming what are referred to as lamellae, or are going to look exactly like the thylakoids, but they're not separate membranes. It's just the cytoplasmic membrane that is pushed in. So you've seen this picture many times in your other textbooks, in other courses. 1, 2 dual membranes, and then here's the thylakoids. If you look at the bacteria, 1, 2 membranes, and then you get these. At first glance, people want to say, oh, look, they're thylakoids. No, they're not thylakoids like you think of with chloroplasts, because if you look right there, the cytoplasmic membrane is continuous with these. So this is actually the cytoplasmic or the inner membrane. 2 membranes, 1, 2, and this is the inner one that's been folded in where it's so much extra of it, has been folded in and around itself. That, fast forward, with a couple 100 million years of evolution, these invaginations, would become separate membranes and thus become the thylakoids we know of in chloroplasts. What's interesting also is that, there are other things out there other than those thylakoid-like things that you're going to find in the green, in the cyanobacteria, or the purple bacteria. You're also going to have what are referred to as chlorosomes. Okay, these are going to be slight pouches, again, of the cytoplasmic membrane. They're going to be in green sulfur bacteria, green non-sulfur bacteria, and phototrophic acidobacteria. So these are not going to be what we think about when we think pigmented photosynthetic, the cyanobacteria, the purple bacteria, the whatever. These guys will have bacterial chlorophyll acting as the antenna, but they're not going to have the reaction center. For them, they're going to be utilizing a central protein known as the FMO protein. You're going to have, the chlorophylls that are capturing, passing on to the FMO. FMO is then going to send it to other proteins that will do the pumping. Reaction center and all those, not quite there. This thing that you're looking at here that you would immediately think, oh, that's another membrane, that's an internal separate. No, these are going to be lipid vesicles, not organelles, okay? Because remember, no organelles, or it would be considered a eukaryotic cell.